Right, hello, you nine, and welcome to another slightly different set of lessons. First off, thank you very much, all of you who have sent in your marks. Um, I asked whether you found it helpful doing these revision, then questions, and then walking, talking, mock, and everyone has said that they have been really helpful. So we're going to be continuing on with that. We're going to be looking, as you can see, at organisation now. So this would have been what we covered after cells in the lockdown. And we're going to do it slightly differently in terms of the revision bit. But there will still be then questions and the walking, talking mock after these. Now, organisation takes in a lot of chapters. So I've actually split this up into 4 parts to make it more manageable and obviously not go into large amounts of time. I'm going to be using a resource that you might use kind of something similar with in other subjects, but it's just a different way of revising, but also I've made sure that it covers all of the specification points. So last set we used the specification for revision. I personally find that really useful when I was doing revision, but this is a different way. Obviously learning different techniques and what works for you is really important. So you can revise for assessments in classes in any of your subjects, but also for the year 11s, for when you get to year 11 and doing the GCSE exams. So what we're going to be using is a resource by a company called Pixel that the school uses. You might have apps and things like that for maths on Pixel, but this is a resource and it's just another way of revising it. So they've broken it down into different sections, and I've put arrows in to what we're going to be looking at today. So this is kind of the overview of what we're going to be doing in the next few lessons. Today we're going to do principles of organisation, and then we're going to do the human digestive system, which comes under the animal tissues, organs and organ systems. Now, there's kind of 2 sections to this resource. The first of all, it will just be revision, so learning it, making sure that we're happy with it, and then there will be some questions afterwards that are to do with what we've just done for you to have a go at and actually apply your knowledge and just having that knowledge recall. So we're going to start with principles of organisation. Now, this is only a small section, so you're not going to want a mind map if you're doing a mind map all on this one, because there's not huge amounts to it. How you do this is not going to be as prescriptive as the last one where we were doing a mind map. If you want to use revision cards, they're really useful, and you would just do a revision card for each bit, just putting the key information. If you want to do it as a mind map, perhaps have it organisation in the centre, and then principles of organisation will just be one of the things that come off it. Or some of you prefer just doing it in your notes. That is also absolutely fine, and just having it broken down in your notes. So we're going to be starting with principles of organisation from the organisation chapters. So what will happen is you'll get a slide like this that contains all of the key information. I'll go through it, but then what I suggest you do is pause it when I've gone through it, and then you transfer it onto whatever revision platform that you want to use, be it revision card, mind map, notes, or anything else. Okay. After we've done this, we'll then do some questions. I'll read through the questions and then you can pause, have a go at answering them, and then we'll go over the answers just to make sure that you do have an understanding of it. So this first section, principles of organisation, is just putting kind of the principles of how we organise things in an organism. So an organism is just a living thing. Okay, now on here we're talking about animals. So an organism for animals is a group of organ systems working together. But a bacteria is an organism, even though it doesn't have organ systems. So it's kind of looking at the whole organism itself, which with animals and most plants will contain organ systems. So examples of organisms: humans, we're an organism. Frogs are obviously organisms, oak trees, orchids, as I've said, bacteria, fungi, all of those we would class as organism. And that's kind of the largest level of organization. Now, in the organism, you will find organ systems. And organ systems are just groups of organs that work together to perform a specific function. So examples in animals and plants, we've got the digestive system. So in your digestive system, we've covered that in a previous topic and it does come up later. You've got obviously the stomach, which is an organ. You've got the small intestine, which is an organ, the large intestine. But all of those together perform a function of breaking down food from large insoluble molecules to small soluble molecules. There's also ones in plants as well, so the root system and the shoot system as well. Now, that's the next level of organization. So in organisms, you find organ systems, and making up the organ systems, you have organs. So an organ is a group of tissues working together to perform a function. So examples are heart, the stomach, leaves, and flowers. So if you think about the heart, for instance, inside the heart, you've got lots of different tissues. It's mostly going to be muscle tissue, but there's also nervous tissue in there. You've got fat tissues in there as well. Lots of different tissues that are performing the function of pumping blood around the body. Now, if we take an organ, that's broken down into tissues. So a tissue is a group of similar cells with a similar function and structure. So muscle tissue is made up of muscle cells. There's lots of those muscle cells that are very similar, they're the same, that come together to perform a function, which is to contract and relax. So examples are muscle tissue, the blood, xylem and phloem in plants. Now, if we take that tissue and break it down into its smallest component, we obviously have the cell, and that is the smallest structural and functional unit of an organism. And we've done different, you have to know about different specialized cells, so we've covered that in the previous topics. But examples include nerve and epithelial cells, but also you've got palisade and root hair cells that are found in plants. So you can see at the bottom how it builds up. You start with something like a muscle cell, lots of muscle cells coming together to form a function. Contracting will be the muscle tissue. Then muscle tissues along with blood vessels, nervous tissue, etc., etc., make the heart, which is the organ that pumps blood around the body. And the heart, along with blood vessels, build up the circulatory system. And the circulatory system is just one of many systems that you find in a human, which is the organism. So what I'd like you to do now is pause this and add information from this that you think is important, particularly things that you didn't know before, onto whatever means of revision you would like to do, be it mind map, revision cards, or anything like that. So if you pause the video now and add that to your revision. Okay, so that is the content, and that does include all the stuff from the specification. So now we need to cover that up, put that aside, and we're going to have a go at some questions. So this will be the question it section of principles of organisation. And there's only 2 questions that I'd like you to have a go at on this. So question number 1 is put the following in order of size from the smallest to the largest. So we've got organ, organism, cell, tissue, and organ system. So from memory, remember, you've covered up your revision notes. Can you put those into size order from smallest to largest? Once you've done that for number 2, for each one, I'd like you to have a go at defining each of the words. So what is an organ? What is an organism? Etc. So pause the video and in your notes, answer those 2 questions. Okay, so hopefully you've had a stab at that, and we're going to go through the answers now to see if you remembered from the revision. So putting those words into size order, the smallest is a cell, and then a tissue, an organ system, and then an organism. So well done if you managed to do that. Trickier one is defining each of the words. So yours won't be exactly the same as this, but words to the effects of. An organism is a group of organ systems working together. An organ system is a group of organs working together to perform a specific function. An organ is a group of tissues working together to perform a specific function. So you can see there's a trend here in words that we need to use. And tissue is a group of similar cells with a similar structure and function. And cell is the smallest unit of an organism. Okay, so it doesn't have to be exactly the same as that, but words to that effect is great. So very well done if you managed that. So we're going to move on to the next bit, and this will have 2 sections to it. So now we're looking at animal tissues, organs, and organ systems, and we're going to be looking at the human digestive system and enzymes, and we're going to be doing these together. Now this will be the end of this lesson after this, because these ones are slightly larger. It will take more time, so don't feel that you have to rush them. We've got 3 slides containing information this time, and then we've got the questions at the end. So we're going to start with the human digestive system, and it is presuming that you know a lot about the digestive system from Key Stage 3. So in Key Stage 3, you do the parts of the digestive system, which you can see on the picture, and have a general understanding that it is there to digest and absorb food, that all of those organs together digest and absorb food. Now for GCSE, you need to know that digestion is where large insoluble molecules are broken down into smaller soluble ones that are then absorbed into the bloodstream. Digestion occurs in the gut. Now the gut is just the whole tube, the whole thing, all the way from the mouth to the anus. And in the digestive system, we use biological catalysts called enzymes that do a lot of the work in breaking things down. So there's 3 enzymes or groups of enzymes that we need to know about. They are listed on the table. We've got amylase, and amylase is a type of carbohydrates, and it's produced in the salivary glands, and it's also made in the pancreas and put into the small intestine. And amylase breaks down carbohydrates into simple sugars. So amylase will break down starch, actually, into a smaller sugar. Protease is a group of enzymes, and they're produced in the stomach and the pancreas. And all proteases break down proteins into amino acids. The last one is another group of enzymes called lipase. And lipase is made in the pancreas and released into the small intestine. And that breaks down fats, fats and oils, which are collectively known as lipids. And they break fats and oils down into fatty acids and glycerol. So I'd like you to pause this slide now. I know that there are links to other parts of it. You can find additional stuff easily on YouTube. If you're unsure about any of this or want to watch a video of it happening, then just search and put in some keywords and you should be able to find it. But can you pause the video and add this bit about the digestive system and starting on the groups of enzymes onto your revision system, be it the cards or the map or anything like that. Okay, so hopefully we have got that down on our revision material, and remember to keep all of these, and I've said in the previous ones, keep all of these revision maps or revision materials ready for year 11 so you can go through them, and it just really helps with revision for year 11. Okay, so we're going to move on to how enzymes works. And again, there is some links to how enzymes work, where you can watch videos on the lock and key theory. And also it is one of the required practicals that from what I remember, Miss Corbett did with you on one of the previous videos. So if you want to have a look at that video on enzyme action, then please feel free to do so. The required practicals are very important. So enzymes, how do they work? They work using a theory that we call the lock and key theory, and it's just a model that we use to show and explain how enzymes break down those large insoluble molecules. Now, enzymes are a group of proteins, and they are called biological catalysts. So they speed up the rate of a reaction. Large insoluble molecules will break down naturally, but it takes a very, very long time. And we don't have that amount of time because we need it in our body to perform lots of different functions. So we use these enzymes, these catalysts, to speed up that rate of reaction. Now, the shape of the enzyme is very important, and where all of the fun happens is called the active site. So that has got a specific shape that fits together with a specific substance. Now you can see there's two diagrams showing the lock and key, and what it is showing is that enzymes will either break things down, such as in digestion, but also enzymes build things up. So when we're actually making our own proteins and our own lipids and our own carbohydrates, we actually need enzymes to put it together and make human ones. So the lock and key model synthesis means to make. So you can see you've got the substrates coming together, joining together, and it's forming one product. In digestion, you've got degradation or breaking down. So you've got the substrate, such as sucrose, that fits together with a specific shaped enzyme and then is broken down into its products. Notice though on both diagrams that at the end of it, the enzyme is unchanged. So that enzyme can go on and break down or build up something else. The products of digestion are used to build new carbohydrates, which I've said, lipids and proteins in the body, and then we use the glucose in respiration in the mitochondria. So if you could pause this video now and add whatever information you want to include for the lock and key theory. Do not just copy out the text because there is a lot of it. We need to reduce it and make it more concise, just taking out key words or combining lots of it into a simple diagram. So pause the video and do that now, please. Okay, so the last thing that I'd like to talk to you about the human digestive system. It covers a couple of things. We've got about enzymes that I'll talk about, and also about the liver, which makes something called bile and why that's important, and stomach acid as well. Now, this is all to do with enzyme activity, which was the required practical. And enzymes are affected by temperature and pH. They have something called an optimum condition, and that's where they're working the fastest they possibly can. That's their optimum condition. If the conditions are not optimums, i.e. if the temperature is too high or the pH is far off, then the enzyme active site will actually change shape, and it means that the substrate won't fit there anymore. And we say that it has been denatured. So never do we say that enzymes are killed, because they are not a living thing. They are denatured, and that's where the active site changes shape. So if we look at the box that starts with enzyme reactions, in enzyme reactions, increasing the temperature will cause the rate of reaction to increase, because those enzymes, those substrates, are moving around faster, so they're more likely to collide. Good link to chemistry and the collision theory. But if it's too high, it changes the active site and they become denatured. Now, if we look in the green box, we've got about pH. So the stomach releases acid, and we know from the diseases topic that one of the jobs of the acid is to kill pathogens. But another job of the acid is to make sure that the conditions inside the stomach are acidic. And that's because the enzymes that are released in the stomach work best in acidic conditions. The enzymes that are made in the pancreas and the small intestine, though, they work better at alkaline conditions. So this is where bile comes into play. So you want acidic conditions in the stomach for those enzymes, but then you need to neutralize it in the small intestine for the enzymes that are released there. And the way that our body does that is it uses this substance called bile. Now, bile is made in the liver, so one of the many jobs of the liver that we've spoken about in a previous lesson, and that bile is stored in an organ called the gallbladder. And it's very alkaline, so it neutralizes the acid, the hydrochloric acid, from the stomach. That's one of its roles, to neutralize stomach acid. It has another role, though, that's very important in digestion. It also emulsifies fats. So that's where you take the large fat molecule and it's broken down into smaller ones, which increase the surface area. Note how many times surface area comes up in biology. The alkaline conditions and the larger surface area means that enzymes such as lipase can break down that substrate much faster. So if you could pause the video now and take out those key points. Again, there's lots of information there. So we need to pick out the key information from that. Try and reduce sections into just a sentence or a picture would be ideal. Can we add that to our revision map or cards, etc. that we're using? Okay, now there's obviously quite a bit that we've covered in these 3 slides. So there's going to be more questions than on the last one we did. So what I want you to do is cover up your revision notes, make sure you can't see them. And can we put down each organ from the digestive system from A to I? So just put A and then whatever that is, B, whatever that is, etc. all the way to I. Now, if you want more time to do that, which you probably will, pause the video. Otherwise, I am moving on to the next slide, the next question. So question 2, what is digestion? So what is the definition of digestion? Number 3, what is an enzyme? Number 4, just sketch a quick table and can we put down what where it's released and what is broken down into what for amylase, protease and lipase. Number 5, what biological molecule are enzymes made of? Number 6, what is the active site of an enzyme? And number 7, what is a substrate? So pause the video and answer those questions now, please. And finally, question 8, what are the products of digestion used for in the body? Number 9, what does denatured mean? Number 10, how does increasing the temperature affect enzyme activity? Remember, there's 2 parts to that. Number 11, how does pH affect enzyme activity? Number 12, what is the role of bile in digestion? And number 13, where in the body is bile made? So pause the video, and if you could answer those last questions, please. Okay, so we're now going to go over the answers to those questions. I would suggest that if you are getting any wrong, that you go have your revision material in front of you and make sure that you have included that information onto your revision map. So if it's not correct, make sure it's on your revision material. Okay, so first of all, we'll go through the parts of the digestive system. So the first part is the mouth. You might have put teeth, I'll accept either. B is the esophagus. C is the liver. D is the stomach. E is the gallbladder, where that bile is stored. F is the pancreas. G is the small intestines. H is the large intestine. And I is the anus. Question 2. What is digestion? It's when large insoluble molecules are broken down into smaller soluble ones. That would be how it is stepped up from key stage 3. We do need to try and remember that definition. Number 3, what is an enzyme? It's a biological catalyst. That's all you need to put. But you might have said that they speed up the rate of reaction. Number 4, we have to complete our table. So amylase is made in salivary glands and pancreas, and it breaks down carbohydrates into simple sugars. Protease is made in the stomach and pancreas and breaks down proteins into amino acids. Lipase is made in the pancreas and it breaks down fats and oils into fatty acids and glycerol. Number 5, what biological molecule are enzymes made of? It's proteins. If you put amino acids, I will accept that because amino acids make proteins, but enzymes belong to the group of biological molecules called proteins. Number 6, what is the active site of an enzyme? It's where the substrate binds to it. Number 7, what is a substrate? It is a substance that the enzyme acts on. So that's what a substrate is. Quite tricky to put into a definition. If you've got something similar, that's absolutely fine, but that is a nice definition to try and remember. Right, last ones. Number 8, what are the products of digestion used for in the body? There's lots of different options. They're used to build new carbohydrates, or new lipids, or new proteins. And a good one to remember because it links with another chapter is glucose is used for respiration. Number 9, what does denatured mean? We must include that the active site has changed its shape. If you then put so the substrate doesn't fit, that's great, but it's the active site changing shape. Number 10, how does increasing the temperature affect enzyme activity? So initially it gets faster or increases the rate of reaction because there's more collisions. But if it's too high, the enzyme is denatured. Number 11, how does pH affect enzyme activity? So you could say that they work quickly if they're in their optimum, but most importantly, if you've said that if it's too acidic or too alkali, the enzyme is denatured. Number 12, what is the role of bile in digestion? There's 2. It neutralizes stomach acid and it emulsifies fats. And number 13, where in the body is bile made? It's made in the liver, and then if you wanted to, or if you have, brilliant, it's stored in the gallbladder. Okay, so that is it from me on this one. So hopefully we have created a section of the revision for organisation where we've looked at the principles and also about digestion and enzymes in animal tissues, organs and organ systems. Hopefully you've had a go at some of these questions as well. As I say, I've broken this whole section down into 4. So the next 3 lessons will be continuing on that. And then there will be some exam style questions for you to answer. And then I'll go through the paper as a walking, talking mock again. Okay, well, have a lovely day. And thank you very much for taking part. I hope you found that useful. Any feedback is always appreciated. So yeah, let me know if you're having any issues on what we've been doing. Have a lovely day, take care, and